welcome to El Zoominario. Today we're very happy to welcome Adriana Romero-Olivares. Adriana is from Hermosillo, Sonora, Mexico, a city in the middle of the Sonoran Desert, a couple of hours south of Tucson, Arizona. She got her PhD in biological science with emphasis in ecology and evolutionary biology at the University of California-Irvine. She is now an assistant professor at New Mexico State University in Las Cruces, New Mexico. Her lab is interested in understanding how microbes respond and adapt to global climate change. Thank you very much, Adriana, for being here.
Thank you so much for inviting me. I'm excited to talk about my research and to share a little bit about my Latinx experience in the US. So I'm going to be talking about a small portion of my research and specifically one project that I conducted when I was a PhD student, but it's been one of my favorite projects, and it continues to inform the research that we're doing now in my lab. So when I decided to study biology, I did it because I was really fascinated by landscapes like this one. So with a lot of visible plants, a lot of biodiversity, and presumably a lot of animals that you couldn't really see, but a lot of plants. There's probably a lot of animals as well. But back then, I never really thought about what was beneath all of these visible biodiversity. But as I moved forward in my degree, I started to learn about the importance of soil and of its very, very large invisible biodiversity and how essential this biodiversity is to keep ecosystems working. So for example, in this image, you can see that in a relatively small piece of land, there's approximately 50 kilometers of mycelium of approximately 500 different fungal species. There's around 100 billion bacterial cells of approximately 10,000 different species. There's hundreds of species of algae, thousands of species of protozoa, and you know where I'm going. There's a lot of biodiversity in this relatively small piece of land that you cannot see with your naked eye, but that it's there. And in my work, I focus specifically on fungi, more specifically on soil fungi, saprotrophic fungi. So what are soil fungi? So soil fungi are fungi that live all of their life or a portion of their life in the soil. And there's a lot of soil fungi. So in a single gram of soil, there can be thousands of different species of fungi. And if you're lucky, sometimes you can see them. So for example, here you can see a piece of wood that has been colonized by a very thick mycelium. So all of these white threads that you can see here, those are fungal mycelium. In this other picture, you can see the type of fungi that we typically think of when someone says the word mushroom. It has like the very traditional cap and it's even reddish. And then here you can see another quote unquote less traditional type of mushroom, but it's still a fruiting body of a mushroom. It's still a fungus and it's still visible to the naked eye. But the great majority of fungi are microscopic their entire life cycle, and sometimes we can see them if we isolate them with microbiological techniques. For example, all these that you can see in the slide, all of those fungi were isolated from desert soils. But there's some other fungi that we have never seen. We know that they exist thanks to molecular techniques. We've been able to see on our computer a sequence of DNA that belongs to those fungi, but we actually don't know who those fungi are. We don't know what species they belong to. We only know that they exist because of molecular techniques, but we have never seen them. And to jump into my research topic a little bit more, as you can imagine, soil fungi, just like any other organism on Earth, they are extremely vulnerable to climate change, to changes in the environment in general, specifically to climate change. But because they're invisible, it's really, really difficult to study them. So for example, in this picture, you can see that the right side of the picture, the land, the ground looks really eroded and unhealthy. You can see that the plant is really struggling. It doesn't have any leaves. And on the other side of the picture, on the left side, you can see the opposite. You can see that the ground is covered by grass. The plant looks pretty healthy. It has leaves. But what did you say about the microbial community in this picture? You can't really say anything. There's things that you can assume. You can probably say that the right side of the picture, the microbial community, is probably not doing great, whereas on the left side of the picture, they're probably doing better. But you really cannot say for sure, because it's really difficult to say something about something that you cannot see. So there's just certain things that for us microbiologists, we simply cannot do with our study system because we cannot see them. So for example, it's impossible to follow fungal life cycle in the soil. It's impossible to count the progeny of fungi. It's impossible to follow the growth rate of a single mycelia on the ground. There's no historical record on the distribution of fungi except for a few species of mushroom, and these records are like very specific to certain areas in Europe. And there's many other reasons why studying microscopic fungi is difficult in ecology and evolution. But despite these difficulties, it is very, very important that we study them because fungi are mediators of biogeochemical cycles. They are responsible for keeping nutrients and elements flowing in our ecosystem, specifically between land and atmosphere. So for example, in this picture, you can see how fungi are mediators of the carbon cycle. So this is a crash course on the carbon cycle. So starting with sunlight and with carbon dioxide, this tree photosynthesizes. And through the process of photosynthesis, it produces biomass in the form of leaves and in the form of wood. Eventually, these biomass is going to die and fall onto the ground, and it's gonna become available for fungi to decompose. So through the process of decomposition, fungi are going to transform this biomass and bound it organically as carbon in the soil. Another portion of this organic matter, fungi are going to use for themselves to build their biomass. And as a byproduct of their metabolism, a portion of this carbon is going to be returned to the atmosphere as carbon dioxide, and it's going to be available once again as carbon dioxide to use in the process of photosynthesis. So you can see that carbon was transformed first, carbon dioxide, then as biomass, then as fungal biomass, soil, and then again carbon dioxide. And the one organism that is in the middle of how, or that is in between these different forms of carbon, are fungi. That's why we say that fungi are mediators of biogeochemical cycles. And there's another amazing group of fungi I do want to point out. These are mycorrhizal fungi, but I usually don't work with them. So in my talk, I'm going to be talking exclusively about free living decomposer fungi. And this is very important because soils are not only the home of soil fungi and of other organisms, but they're also a carbon storehouse. What does this mean? So there's around twice as much carbon stored in the soil compared to what's already available in the atmosphere. So because soil is a storehouse of carbon, it can also be a source of carbon dioxide. And it is for the best that carbon stored in the soil stays in the soil, because if it's returned into the atmosphere, it's going to be returned in the form of CO2. So in a few words, if a lot of the carbon that's stored in the atmosphere, in the soil, is released into the atmosphere as CO2, the greenhouse gas effect and thus global warming would worsen. And all of this is really important because the molecular machinery that microbes use to decompose and store and return CO2 into the atmosphere is extremely sensitive to environmental change. And in fact, impacts to soil fungi, so any change in the environment can impact soil fungi And it can result in all sorts of different things. For example, it can result in millions of dollars lost in crops. It can result in increases in greenhouse gas emissions, as I just mentioned, and it can even cause changes in the microbial community that could result, for example, in the emergence of pathogens of plants and animals. So with all of this in mind, I went to the boreal forest in Alaska. And I was especially interested in studying this ecosystem because boreal forest stores approximately 510 billion tons of carbon in the soil. And changes in temperature brought by global climate change has been really, really drastic in these latitudes. So in this map, you can see the location I work in. It's known as Delta Junction right here. And according to this map, so this map is a map of the state of Alaska. All of the state of Alaska, including the area of Delta Junction, is significantly affected by changes in temperature. You can see here all of the changes in temperature from 1969 to 2018. And specifically, the area of Delta Junction has experienced an increase of 1.7 to 2.2 degrees Celsius in the average annual temperature compared to the past 50 years. So I conducted research in this field experiment. At the time I sampled, the experiment had been ongoing for over 10 years, and it's a really simple experiment. It consists of 5 greenhouses in the middle of the forest, just like this one in the picture. And in the other picture here, you can see where each one of the greenhouses was. And the way that this experiment works, or the way that the greenhouses work, is really simple. These greenhouses passively warm the soil and the air inside them. And they increase the inside air temperature by 1.5 degrees Celsius. So around the increase in temperature that this area is actually experiencing in. And it increases the temperature of the top 10 centimeters of soil by 0.5 degrees Celsius. Evapotranspiration is higher inside the greenhouses, and this causes that the inside of the greenhouses is 22% drier compared to the outside. And something that I hear very often is, 1.5 degrees Celsius is really not that hot compared to the outside. How can that possibly be affecting the plants and the microbial community inside of the greenhouses? But here you can see how different the inside of the greenhouses looks compared to the outside. So you can see how the outside of the greenhouses look. So these are basically just the space outside of the greenhouses. So these are the control plots, which were usually situated just a few feet away from the greenhouses. And then you can see how the inside of the greenhouses. So this looks extremely dry. Here you can see multiple mushrooms all around here. And this one, it's really dry, hardly any moss. There's one mushroom there, but there's the contrast is really strong. So I wanted to know what's happening with the soil fungal community under warming and drying. Where are they allocating resources, and how does this reflect in the cycling of carbon? So to answer these questions, I carried out soil metatranscriptomes. This is basically just extracting RNA from control and from treatment plots, and then analyzing those transcripts for metabolic maintenance genes, also known as COG genes, and decomposition genes, also known as Casii genes. And I wanted to analyze soil metatranscriptomes because that way you can get a sense of what the fungi are doing in that exact moment that you sample. And I also wanted to measure decomposition rates. And to do this, I used a very simple experiment known as decomposition litter bags. And the way that this works is that you have litter bag, which you can see here. You put in the litter bag a specific amount of biomass. In this case, I use spruce needles because spruce are the plants most common in this site. And then you just put a specific amount of biomass, deploy it on the forest floor, and then let decomposition happen. And after a few months or years, you collect the litter bags, you weigh them, and then based on biomass loss, you get a sense of how much decomposition has happened. So what did I find? So I found that warming and drying favors melanized, pathogenic, and desiccation tolerant fungi. So these groups of fungi were the ones that were more active under warming. And by more active, I mean the ones that were transcribing the most. So basically a lot of black fungi like this one, Silaria, all black fungi are melanized fungi. They produce a lot of melanin, which is a pigment, which confers them stress tolerance. I found a lot of crust fungi like this one. These fungi are also very drought tolerant and stress tolerant. A lot of pathogenic fungi, specifically plant pathogens, like fungi that belong to the order of the Diabortales, and a lot of fungi that belong to fungi that are pathogenic when conditions are correct, like penicillin, for example. And then a lot of lichens too, which are also very drought tolerant. In addition, I found that metatranscriptomes revealed a trade-off in resource allocation. So in these graphs, in the x-axis here, you'll see metabolic genes, and here you'll see decay genes. And then in the y-axis, you'll see the total number of gene transcripts. And then white or grayish are the results for control plots, and then black are the results for warm plots. So as you can see, the trends are the exact opposite, meaning that under warming, microbes were allocating more resources to cell metabolic maintenance and less resources to decay, whereas in control conditions, you'll see the opposite. So in very straightforward terms, what does this mean? So it means that under warming, fungi were basically stressed, and they were investing a lot of resources in just basically staying alive. And because they were investing a lot of resources in staying alive, they didn't have enough resources to allocate into decomposition. And I do want to point out that transcripts are just that, they're transcripts. You cannot be sure that all of these transcripts are going to mature and are going to be translated into enzymes. And we also cannot be sure that these changes will have an impact at the ecosystem level. However, our field decomposition experiment supported my metatranscriptome's findings. So here in this graph, in the x-axis, you'll see decomposition time, and in the y-axis, you'll see mass remaining. In this case, control is white and warming is black. And what you can see is that for all collection times, the percentage mass remaining was always higher in warm plots. What does this mean? This means the decomposition in warm plots was lower compared to control plots. So you can see that every time there was less biomass remaining in control plots compared to warm plots. And it's a little bit counterintuitive because you see more here. So then you say, oh, so then there's more decomposition under warming. No, it's actually the opposite. You see more here because there was more mass remaining, meaning that decomposition was happening at a slower rate. So to answer the questions that I had, what's happening with the soil fungal community under warming and drying? Pathogenic, melanized, and desiccation-tolerant fungi were more active under warming. Where are they allocating resources? They're allocating more resources to cell metabolic maintenance and less resources to decay. So remember, the fungi are basically stressed, trying to stay alive. They're allocating a lot of resources to staying alive, and they cannot afford resources to decay. So how does this reflect in the cycling of carbon? There's less decomposition under warming. So questions that I get very often are these results good or bad? And the answer is that it's a little bit of both. So first, it looks like it's good because less CO2 is potentially being returned into the atmosphere. So remember, less decomposition, less CO2. But the shifts in the fungal community are really concerning, especially the increase in pathogenic fungi. And we also have to remember that there's a lot of microorganisms and plants and other organisms that acquire their nutrients from fungal decomposition of organic matter. So less decomposition results in less nutrients available for plants and animals as well. I do want to point out that these results are not unique to our field experiment and that others have found very similar results. And it's too soon to tell. But what we know is that global climate change is likely to affect the fungal community with consequences at the ecosystem scale, such as disruptions to the carbon cycle. Oh, I just wanted to point out that these are things that I looked into. So the fact that other studies have found very similar results, it's something that I reviewed in this meta-analysis. And I do want to point out that in the decomposition study that we did, there are some nuances, which it's too technical, and I'm not going to go into it. But there's a specific type of carbon that it's being decomposed a little bit faster under warming compared to control conditions. So to summarize what I just showed, global climate change is affecting fungi from genes to ecosystems. I hope that I convinced you that fungal responses to environmental change are impacting biogeochemical cycles. I hope that this was obvious to you based on my presentation, and that fungal responses to environmental change are disrupting the carbon cycle. I also hope that I've convinced you that studying fungal responses to global climate change allows us to understand mechanisms behind ecosystem scale observations. With that, there you go. I want to thank you, and I want to thank all of my funding sources that supported this research, the University of California, CICESE, which is a Mexican research institution, the JGI, CONACYT, which is the organization that provided with my fellowship for me to conduct my PhD in the US, and tons of different funding sources from both the US and Mexico. My PhD advisor, Kathleen Cedar at UC Irvine, my collaborators in Mexico, and my amazing undergrads during my PhD, John No, Diana Nguyen, and Hao Trong. And with that, I'd be happy to take any questions.